Hi, I'm Shuze. I'm a third-year PhD student at the University of Michigan. Today, I'm going to talk about molecular mechanism conferring spatial identity during cochlear duct extension. Hearing is mediated by a specialized sensory epithelium, the organ of Corti, which is located in the cochlea of the inner ear. The organ of Corti is stretched along the apex to base, tonotopic axis of the cochlea. High frequencies are detected in the base, while low frequencies are detected in the apex. Generally, frequency tuning of hair cells requires a specific set of proteins to be expressed, with tonotopic gradients. Therefore, we are interested in identifying the molecular mechanism conferring spatial identity during cochlear development. During embryonic development of mice, a group of primordial cells at the base of the cochlear duct divides, and thereby contributes to the elongation of the organ. Around embryonic day 12.5, apical cells are born first, and during the following 48 hours, new cells continuously elongate the duct until the final size is reached. A cross-section through the cochlear duct at E14.5 highlights the asymmetric organization of the cochlea. Therefore, we proposed two hypotheses of cochlear extension mechanisms. First is time-space translation model. Time-space translation model is strictly controlled by time in the organ of Corti development, which translates developmental time into spatial information. Specifically, the apical cells of E12.5 data is similar as the apical cells of E14.5 data, and the basal cells of E12.5 data is similar as the mid-basal cells of the E14.5 data. And the second model is called dynamic morphogen model, which morphogen-based signaling originating from the opposing ends of the cochlear duct could potentially confer spatial information. Specifically, the E12.5 apical cells is similar as the E14.5 apical cells, and the basal cells of E12.5 is similar as the basal cells of E14.5 data. Based on the hypothesis, our goal is to understand the mechanism that characterizes the spatial identity during cochlear duct extension. To achieve our goal, we analyzed the single-cell RNA-seq data of the developing cochlear duct across two different time points, E12.5 and E14.5. Here, we summarize the cell number for each time point, and we have two replicates for each time point. Next, we identified clusters and annotated cell type for each dataset. By concentrating cochlear duct cells, including cochlear floor and roof cells, we successfully reconstructed the 2D and 3D original structure of the cochlea. Specifically, from the 2D projection, the x-axis distinguishes the subpopulations of the cochlea, and the y-axis corresponds to the tonotopic axis. The circular projection resolved the asymmetric organization of the cochlea. To reconstruct the cochlea in 3D space, we projected cells onto the surface of the cylinder, with the height of the cylinder representing the tonotopic axis. Also, the reconstruction specifies the three major axes of the cochlea, including roof floor axis, medial lateral axis, and apex base axis. Take E14.5 dataset as an example. OC90 and LUM characterize the roof and floor cells. FGF10 and BMP4 distinguish the medial and lateral side of the cochlea. And FST and MKI67 are the gene markers representing apex and base. To test our hypothesis, conferring spatial identity during cochlear duct extension, we focused on the cochlear floor cells and first identified gradually expressed genes along the tonotopic axis using KL divergence. Here we showed overlapped gradually expressed genes between E12.5 and E14.5 datasets using heat maps. Each column is an individual cell ordered along the base to apex axis. Each row is a gradually expressed gene colored by gradient direction. The purple color indicates the genes that are highly expressed in apex, and the yellow color represents the genes that are highly expressed in the base. Overall, the gradually expressed genes characterize the cell type identities. To individually visualize the gradually expressed genes, we screened 4 candidates. We ordered the cells along the x-axis from base to apex. HMGA2 and FST are 2 gradually expressed genes with decreasing levels from the apex to the base. HMGB1 and HMGB2 have the opposite gradients along the tonotopic axis. Next, we use the gradually expressed genes as features to test our hypothesis. We aggregated 10 cells as a metacell and calculated the similarity matrix with Gaussian kernel for pairwise pentacells between E12.5 and E14.5. Heat map were used for visualization. Both columns and rows are metacells ordered along the tonotopic axis. From the heat map, it shows that the basal E12.5 cells is aligned with the basal cells of E14.5 data. Therefore, it supports the dynamic morphogen model. To test the robustness of our findings, various parameters were used to generate the affinity matrix were adjusted. The results are consistent across the various parameters. In summary, the findings strongly support the dynamic morphogen model. In summary, we resolved the original structure of the cochlear duct in 2D and 3D space, and also we identified the molecular mechanism conferring spatial identity during cochlear duct extension. And thank you for your listening.